A genome sequence is useless without annotations. When we do genome annotations, we are looking into there are three major steps in this genome annotation. Number one, we try to find features which are not associated with the protein coding genes. For example, we are looking for something like transfer RNAs, ribosomal RNAs, small nuclear RNAs, we call snRNAs, signs, lines, and microRNA precursors. So these kind of things are the features which are not associated with protein coding genes. We try to build models for protein coding genes also in annotation, including exons, finding coding regions, and regulatory regions. The third step is we try to associate biologically relevant information with the genomes. So these things are three important components of biological annotations. Methods of these annotations. So in computational sense, we classify them into following methods. One of them is ab initio methods. They are based on sequence alone. So for example, we try to do some gene finding, and that is dealt by gene prediction algorithms like Augustus, Glimmer, GeneMark. We try to find the repeat families. So there is software called as repeat masker. So they try to work on the sequences, and they try to work finding these different genes and repeats based upon some specific patterns. There are evidence-based methods. So we try to hunt the genes through their messenger RNAs. We can take the transcriptome data and then align the cDNA. We take that messenger RNA, we get the cDNA, and then we try to assemble the genomes while using these cDNAs. So these cDNAs, they are coming from the genes. So when we put them together, when we make their assembly, we can try getting and identifying the genes. There are different softwares like Top Hat, Cufflinks, etc. We'll discuss when we talk about gene expression or the analysis of next generation sequence data. We'll take these softwares over there. When we are trying to do those biological annotations, we try to actually infer some biological meanings. So in that case, what we can do here is, number one, we can blast those genes against protein databases, and we can have the sequence similarity, and from those sequence similarities we can identify these gene models. They can produce what kind of protein. So in this way we can identify their proteins. An example is we have InterPro-scan, which we can do here is that we can take the proteins and we can compare them. We can get different patterns from those genes. While we take those genes and we try to identify different protein patterns which are present in those genes. So in this way we can take those databases of protein domains and patterns and use them. We can also find out the gene ontology terms, which are called as Go terms. One of the example is BLAST2Go. There is a software. So in these gene ontology terms, what we do is we try to see what are the functions of those genes. So much of the data processing in bioinformatics involves searching and recognizing certain patterns within the DNA. So whatever you are doing, whether you are trying to find those motifs, whether you are trying to find those domains, or you are trying to do some exact searches, so in that case you are actually doing these pattern finding things. So in biology it means finding a motif within a DNA or within a protein, whereas in computational sense, pattern finding is a job where we try to search a pattern within a string. So what we have seen in this section is that the genome, after it is assembled, genome annotation is an important step, and we try to find different patterns and we try to find different functional aspects of the genes. So yeah, as far as this job is concerned, pattern finding is an important aspect in genome annotations.